All right. Hello, everybody, and welcome to the afternoon sessions. Quite blinding, the lights here. So you'll hear 4 speakers today on imaging brain plasticity, ranging from diffusion to structural MRI. And very much appreciative so many of you coming. Looking forward to the talks. And the first of them will be given by Heidi Johansen-Berg from Oxford University. I'll turn it over to Heidi.
So thank you very much. I'm going to focus in this talk on ways in which we've looked at plasticity, both functional and structural, using high-field MRI. And I'll start off by talking about some examples where we've exploited the capacity of 7 Tesla human MRI to be able to do high-resolution functional mapping. And one of the first things we tried to do when we installed our 7T MRI scanner in Oxford was test whether we could use it to pick up individual digit representations in the human brain. So this is work from James Kolosinski when he was a PhD student in the lab, where he optimized a phased encoding paradigm, where, as you can see, as the subject moves in sequence the fingers of their hand, you see a traveling wave of activity moving up the central sulcus, such that if you then classify each voxel along that wave of activity according to its preferred finger, you can derive these very consistent maps of individual digit representations, where you can see this clear topographic gradient such that the index finger is represented inferiorly, moving up to the little finger more superiorly. And by scanning a number of different individuals over different time points, James was able to show that these maps are highly variable between subjects, but highly reproducible within subjects. So here, for example, you're seeing maps from 3 different individuals, where you see that that same topographic gradient running from pink to blue as you move up the central sulcus is preserved across all of those participants, but the precise shape and size and distribution of those maps varies somewhat between participants. But if you then bring people back on different days to perform the same mapping again, you can see there's a remarkable degree of reproducibility within subject over time. So this then provides a really nice starting point from which to ask the question, under what circumstances can we change those maps? If we alter subjects' sensory motor experience, do we then see any changes in these map organization? So to ask that question, James performed this experiment where subjects came into the lab on a number of different occasions, and we compared scans acquired before and after 2 different 24-hour periods. In one, the control period, the subjects behaved as normal, and then in the other, he glued together their index and middle finger with a surgical glue, a removable surgical glue, for 24 hours. So for 24 hours, those 2 fingers were fused together, and we then tested to see what effect that altered sensory motor experience would have on the cortical maps. For those of you who are familiar with the non-human primate literature, you'll know that there have been many classic monkey studies done with similar types of manipulations, but over much, much longer timescales. And what they would predict is that if you fuse together digits 2 and 3, as we did, then you should have a moving closer together of the cortical maps of digits 2 and 3, where these circles are a schematic representation of the cortical maps. So this is what we might predict from the monkey literature, but here we're changing experience for a much shorter period of time, for just 24 hours. And what we found was that 24 hours of altered experience produced quite a different change in the pattern of those cortical maps. So here on the y-axis, we're plotting the amount of overlap between the maps of different digit pairs, and you can see that for the different experimental conditions. So you can see the overlap is stable between the control conditions, as you'd expect. But what we're seeing is, interestingly, no change in the bottom line, which is the overlap between the digits that are glued together, but instead a change in the other digit pairs, such that there's less overlap between digits 3 and 4 and more overlap between digits 4 and 5. So in other words, what's happening after 24 hours is something more like this. As you glue together 2 and 3, 4 and 5 move closer together, and 3 and 4 move further apart. So this interesting neural observation gave rise to a new behavioral prediction. So you might expect that the degree of cortical overlap between different digit representations should have some consequence for how well people can discriminate tactile inputs to those digits. You might predict specifically that for the digit pair that has increased cortical overlap, 4 and 5, subjects would be worse at differentiating tactile inputs to those digits. Whereas for the digit pair 3 and 4 that have less cortical overlap, you might expect better ability to discriminate tactile inputs to those 2 digits. So we tested that behavioural prediction in a new experiment where instead of scanning subjects before and after the gluing manipulation, James tested them on a temporal order judgment task where you deliver pairs of tactile stimuli to pairs of digits and you vary the interval between those stimuli such that you can come up with a score which tells you how good subjects are at discriminating tactile inputs to pairs of digits. And what we found was that the behavioural change fits the prediction from the neural change. So here I'm just reminding you what the neural result was in terms of the cortical overlap. And then here I'm showing you the behavioural results, where now on the y-axis we're plotting that discrimination score came from the temporal order judgment task, where higher scores represent better discrimination. And you can see that for the digit pairs where we have reduced cortical overlap, we're getting better capacity to discriminate between those inputs, whereas for the digit pair where we're getting more cortical overlap, we're getting worse ability to discriminate between those inputs. So what this shows then is that in healthy intact subjects, if you alter somatosensory experience for a brief period of time, just 24 hours, you can change the precise characteristics of these cortical maps in a behaviourally relevant way. But it's important to point out, though, that certain organisational principles of those maps are relatively preserved, are stable. So we still have the same topographic gradient moving from index finger to little finger as you move up the central sulcus. So one question we had then was, what if we had a much more dramatic change in sensory motor experience? What happens, for example, in the brains of people who've had an upper limb amputation? And this is work done by Tamar Makin in the lab, where she's interested in how limb amputation alters brain representations. And a prevalent model of what happens after limb amputation is that you remove the input to the hand area and then neighbouring cortical representations, such as the face, invade that freed up territory. But Tamar's taken a slightly different slant on this, and what she's done is tried to access the representation of the missing hand. And to do this, she asks amputees to move their phantom limb. So many amputees have a very vivid phantom limb. They can move that limb, they can feel that limb. So if you put them in the fMRI scanner and ask them to move their phantom, the activity patterns they produce, indiscriminable, are the same as activity patterns in intact people moving their intact limbs. And that's shown here in terms of cortical maps, where you can see the activity is the same for people moving a phantom limb, which isn't there, versus moving an intact limb. And you can see that quantitatively here in an ROI analysis, where you see that there's no difference between the amputees and the two-handed controls. Whereas if, for example, we take one-handed individuals who were born without a hand, born with only one hand, who do not have phantom limb, if we ask them to move their missing hand, they think it's a very silly question, but if they try to move that missing hand, they don't activate the same area of the cortex. So there's preserved representation of the phantom hand, but another question is, how organised is that representation? And to address that question, Saneh Kikert, a student of Tamar's, used that same high-resolution digit mapping procedure to test whether that topographic organisation of the digit maps is preserved even decades after losing a limb. So here you see two control subjects from Saneh's study, where you can see that same topographic gradient that I described previously. We're running here from the thumb up to the little finger, moving inferior to superior up the central sulcus. So just like we showed before, this very consistent topographic gradient in controls. Saneh then found two very unusual amputees who had incredibly vivid phantom sensations, such that they could then individually move the individual digits of their hand, despite that hand having been amputated 20 or 30 years previously. And when we look at the fMRI data from those amputees, we find that preserved topographic representation. So here you see again that same gradient of activity moving from digit 1 up to digit 5. So together these results shows that we can rapidly change the fine details of these maps with experience manipulation in intact subjects, but nevertheless some of their organisational principles, such as these topographic gradients, are very highly preserved after even decades of altered experience. So then for the second part of the talk, I want to tell you about some of our work on structural plasticity, and particularly white matter plasticity. So we're interested in whether the brain's white matter fibres can alter their structure with experience and training. And our first observation here was some years ago from Jan Scholz, a student in the lab, who showed that when healthy human adults learn a new visuomotor skill, in this case juggling, we see changes in their brain structure. We see increases in grey matter volume, shown here in the red clusters, but also increases in the fractional anisotropy of underlying white matter fibre pathways. So this was interesting because these were relevant areas of the brain for this particular task, which is a visuomotor integration task. It was also interesting because this was the first demonstration of experience-dependent white matter plasticity in humans. But it was frustrating because we don't know what these changes in the white matter represent in terms of the underlying cellular structure. And here are just a few of the many, many potential interpretations of what's going on here. So changes in fibre organisation or myelin, any number of different alterations could give rise to an increase in anisotropy. So to try to tease apart some of these different possibilities, we've carried out parallel studies in rodents in which we can not only take diffusion measures using imaging, but we can also then follow up with cellular level investigations. And this is work done by Cassandra Sampao-Baptista. So what Cassandra did was train rats to learn a new motor skill, in this case reaching for a food pellet, and then she scanned the rat brains post-mortem and compared the anisotropy maps in the trained versus control groups and found the result that you see here, which is the red cluster in the white matter underlying the forelimb motor cortex, where there's increased anisotropy in the trained group relative to the control groups. So this, if you like, is analogous to what we saw in our human jugglers. But then because this was an animal study, she could then go in with immunohistochemistry, here staining for myelin basic protein, and show that that same area of white matter has greater myelin in the trained animals compared to the control animals. More recently, we've been carrying out longitudinal imaging using a similar paradigm here in collaboration with colleagues in Utrecht in the Netherlands, and adding MTR imaging, which should give us a more myelin-specific metric. And you can see a similar pattern here, that if we compare images taken before and after training, we see increased MTR in the trained group compared to control groups. One of the many questions that arise from these observations is how quickly can activity-dependent myelination occur? And a couple of years ago, there was this intriguing paper that came from Stanford suggesting that it could occur potentially incredibly quickly. So what this group did was use optogenetic stimulation to drive neurons in the premotor cortex of the rodent brain, and then they looked at the proliferation of oligodendrocytes, the myelin-forming cells in the underlying areas of white matter. And what they found was that just half an hour of increased activity gave rise to dramatically increased proliferation of oligodendrocytes. So gave rise to events that could then result in increased myelin down the line. So this suggests that even quite brief periods of focused, altered activity could give rise to white matter change. We wanted to explore the timescale of activity-dependent white matter change in humans. You could do this, for example, through other types of brain stimulation, but we chose instead to use neurofeedback, fMRI neurofeedback at high field, which allows us to tap into the brain's own intrinsic capacity for activity modulation. And this is work done by Cassandra along with Heather Niedli. And so in a neurofeedback setup, you have somebody lying in the fMRI scanner, as shown here. We record signals of their brain activity, and then we feed back some visual representation of that activity, where the subject is instructed to modulate that visual representation in some way which corresponds to modulating their brain activity in a particular way. So this provides us with a way to modulate activity in a targeted brain region and then test what effect that activity modulation has on brain structure. So subjects came in for a number of different scans, and on 2 scans they performed a neurofeedback condition, 2 different neurofeedback conditions. It's a very simple experiment. All the subject has to do is move their left hand, but they do that while looking at the visual display, which displays 2 coloured bars whose height represents activity in the ipsilateral or contralateral sensory motor cortex. And in one condition, subjects are told to increase the size of both of those bars. In another condition, they're told to decrease the blue bar while increasing the red bar. So in other words, what differs between these 2 conditions is what they're told to do with the ipsilateral sensory motor cortex, such that in one they're instructed to send it up, and in the other they're instructed to send it down. So we can look first of all at the fMRI data acquired while they were performing this task to see whether they were effectively able to use the neurofeedback to modulate their brain activity, and we found that indeed they were. So this is a ROI analysis plotting the degree of activity in those 2 different regions of interest, where if you focus on the ipsilateral hemisphere in blue, you can see that as instructed, in the IPSI up condition, which is the white bars, they're increasing activity, whereas in the IPSI down condition, which is the grey bars, they're decreasing activity. Whereas for the contralateral hemisphere, where the instructions don't differ between conditions, there's no difference in activity. So then the question is, to what extent does this modulation of activity in the ipsilateral sensorimotor cortex alter brain structure? And to address that question, we can look at DTI scans acquired before and 24 hours after each of those neurofeedback conditions to look for a condition-by-time interaction. So to ask, are there any areas of the white matter where there's a change in anisotropy pre to post neurofeedback that differs between those two conditions? And we do find such an interaction in the cluster here, in the corpus callosum, such that we're detecting a change in corpus callosum anisotropy 24 hours after activity modulation. And if we unpack the data that are underlying that interaction, they're displayed in the scatter plot here, where you can see that that cluster is showing increasing anisotropy in the increased IPSI condition and decreasing anisotropy in the decreased IPSI condition. So in other words, by modulating activity in a targeted brain area, we can then detect changes in white matter microstructure within 24 hours. This raises many different questions, not least of which is how quickly has this change occurred, whether this change has anything to do with myelin. So obviously, we're not directly measuring myelin here, we're measuring anisotropy. And those are all questions that they're following up on currently. So in summary then, high-field MRI offers sensitivity to structural and functional plasticity, and the examples I've told you about today show that rapid plasticity of digit map overlap can be detected after altered experience in just 24 hours, but that there's preserved topography of those digit maps even years after limb amputation. And then finally, that we can detect rapid changes in white matter microstructure after targeted modulation of activity. So finally, just want to thank all of my collaborators and colleagues, as well as my funders, and thank you very much for your attention.
We have 3 minutes for questions. Please come up to a microphone.
I had a question about, sorry.
Oh, yeah.
Yeah. I had a question about FA changes you reported in your last study. And in general, I was curious about whether the FA changes are driven by MD or axial diffusivity versus RD, and whether you see any specificity or differences in the time course of what is driving the FA changes.
Nothing particularly surprising in that study. So we see similar effects in MD and in RD, but there's nothing, unpacking the different components of the tensor don't really help to shed much light on what could be driving those effects. But yeah, it's the pattern that we very often see, which is that you see co-localized decreases in MD and decreases in RD in the same location.
So our next speaker on the tour of imaging brain plasticity in the brain is going to be Robert Zatorre from McGill University and the Montreal Neurological Institute to tell us about predisposition plasticity in auditory learning. Robert.
It says I have 4 minutes. It's going to be a short talk. There we go. If all of us are frowning when we're up here, it's not because we're really upset. It's because the lights are right in our eyes. So thank you very much, Jason, for organizing this lecture. My talk is actually follows rather nicely, I think, from what Heidi has told us. But I want to emphasize a couple of other features about plasticity. One is the idea of predispositions, and the other is about hemispheric asymmetries. So the usual paradigm that all of us use to study plasticity goes something like this. You start with the brain, then you perform some kind of manipulation, training of some sort, or whatever you're interested in, and then you look for changes that occur, and those changes are presumed to be causally related to the intervention. But when we're looking at something like high-level skills, I would argue that there might be some interesting effects that would happen, so that if the brain that you start with belongs, say, to this fellow, then after you give this individual some musical training, you might end up with a very special kind of outcome which might not be the case for every individual. And so that's one of the concepts that I'm particularly interested in. This concept plays out in the following way. When we look at the sort of model system that we're interested in, because we're especially interested in musical training as a way to understand plasticity, and the brain networks that are involved are sort of shown in this cartoon here, involving both connections between auditory and motor regions. We don't have a pointer here, do we? No, we have a pointer, but it's hard to point. Wait, is there a cursor? There's a cursor, but it doesn't correspond to this thing. Oh well. So if we're interested, one of the nice things about musical skill is that we can implement it in a nice experimentally controlled way. And so this is what was done in this project run by Sibylle Herholtz and Emily Coffey. And we were interested in longitudinal training. So we took a group of people who had no musical background in particular, and we performed 3 functional scans on them. Between scan 1 and scan 2, there was a baseline period, there was no training, and when we looked at the brain activity, there was no real significant change going on. But then in the second time interval of 6 weeks, the participants received this kind of training. So every day, 5 days a week, for 1 hour a day, they had to perform these progressively increasing difficulty tasks where they would listen to a pattern of tones. They would then see on the screen the corresponding movements. They would then have to imitate them by playing on a little keyboard, and then they would get feedback about the correctness of their performance. And this allowed us to look at how well people learned, and allowed us to see, to test what would happen in between the 2 scans corresponding to before and after the training. Now, in the scan session itself, there were 2 tasks that were performed. These tasks involved presentation of the melodies that were part of the learning. They were actually familiar melodies. And there were 2 conditions. The 2 conditions aren't so relevant for the training, but briefly I'll tell you that one was a listen condition where people heard the melody, and then at the end there was a probe tone that was either correct in pitch or not. And there was also an imagery condition where for the first several seconds they heard the cue. They then had to imagine the continuation of that melody, and then again there was a probe tone, and they had to say if it was the correct pitch or not. Behaviorally, people were able to do this, indicating that they were able to imagine the melody. So when we compare scan 3 to scan 2, and we look for changes associated with the training, while people are merely listening, so they're listening to the identical stimuli in both conditions, and what we see is then both in the imagine and listen condition, we see very clear recruitment of the more dorsal component of the auditory motor system, in particular involving the dorsal premotor cortex and the interparietal sulcus. And I noticed that on the word cloud there that was up before the session started, the word reproducibility was one of the big words. And as Heidi also mentioned, it is indeed possible to obtain very consistent data across quite different conditions. So this is a nice feature. Our interpretation of this finding is that it represents some kind of enhanced interaction between the auditory and the motor systems, such that when you've learned to play these melodies, particularly the dorsal component of this system is engaged even when you're merely listening, because you now have a motor representation that's associated to the auditory representation. So that's the sort of plasticity account. But we also noticed something interesting when we looked at the behavioral performance during the training time. So these different colored curves here represent different individuals and their ability to perform the task as a function of time. And as you can see, there are some people who really learned quite well, others who were intermediate, and others who really were quite poor. And we wondered if this degree of variability could somehow be captured in the brain activity pattern that was associated with the pre-training scan. So recall that in the pre-training scan, they're just listening to melodies. They haven't yet learned to play them. They're just listening to them. And so we asked the question, if we use the slope of that learning curve as a regressor, can we see any brain activity that would be predictive of that performance obtained subsequently? And we obtained actually two regions. The main one I'm talking about today is in the right auditory cortex. There's also a hippocampal region. And this is quite interesting to us because it is telling us that something about the brain activity profile in the right auditory cortex is such that prior to training, those who have a more enhanced activity are the ones who will later go on to learn more quickly than those who have less activity. The right auditory cortex is a favorite region of mine, and we've been seeing it consistently in many different studies. So, for example, in the study done by Nick Foster some years ago, we looked for structural correlates, and we found them both in terms of VBM measures and cortical thickness, so that these structural features were correlated with performance on a pitch task. So we think there's something interesting going on in the right auditory cortex. It seems like some properties of the auditory cortex are relevant for auditory motor learning, but we wanted to see if we could find more direct evidence for some kind of interaction between the right auditory cortex and the motor system at the network level, because in the previous study it was really just at the sort of univariate level. So to do this, one of my former postdocs, Jamila Ando, was interested in using TMS to see if we can look sort of directly at the interactions between auditory and other systems. And so she developed this technique where we apply continuous theta burst stimulation, which is an inhibitory sort of TMS protocol, to the right or left auditory cortex. And we obtain resting state data before and after this manipulation, allowing us to look for the differences in the relevant brain networks that occur after the TMS. So when we look at the effect of TMS on the right side, so we're applying TMS to the right auditory cortex, we see a modulation in two different networks. One is the auditory network, and we see an inhibition primarily contralaterally, also to some extent ipsilaterally. And this is sort of makes sense. You stimulate auditory cortex, you get a change in the auditory network. But we also saw, under the same conditions, that there was a significant change in the somatomotor network. And in particular, there is a change bilaterally in the dorsal premotor regions and in the supplementary motor area. This did not seem to happen with stimulation of the left side. We were unable actually to see any significant change under the conditions of testing in any network. So there seems to be a big asymmetry going on here, and also something demonstrating interactions between auditory and motor systems. The degree to which this interaction occurred in the auditory network, at least, was dependent upon anatomical features of the corpus callosum. So if we extract the tract that interconnects the left and right auditory cortices, the degree of inhibition we observed contralaterally was related both to fractional anisotropy and number of streamlines in that tract. This does not happen when you look at a control region like the visual cortex. So it seems like there are important individual differences in the degree to which these networks are modulated by the TMS. So we see evidence from the study of some kind of preferential connectivity between the right auditory cortex and the motor system, which is modulated by individual differences in anatomy. And so we also wondered whether we might be able to find more direct evidence of structural network asymmetries. And that's sort of the theme of this symposium, is that there are structural network properties that might be very important to understand in terms of plasticity. So not knowing very much about structural network types of analyses, I turned to my colleague Bratislav Misic, who knows quite a lot about this. And I asked Bratislav, well, would it be possible to look at what's going on in network properties, particularly if we are looking at the way that these networks are different between the right and the left auditory cortices. So to explore these questions, Bratislav has come up with 3 different measures. These, the first 2 are really fairly well known, I think. The first one is just direct path length. So if you start at one particular node and you look to see what is the shortest path to get to any particular other node, that would be the path length, which is kind of an efficiency metric, if you will. Then there's another one called communicability, where you take into account not only the direct pathway, but also all the other possible pathways between a particular source and a particular target, and you weight those according to the number of steps. So that's so-called communicability. And there's a third metric, which I find particularly intriguing. This is one that Bratislav developed when he was working in Olaf Sporn's lab. And this is a technique where you basically introduce a perturbation in a particular node, and then you look to see what is the propagation of that perturbation across the network, and you measure the amount of time that it takes for the perturbation to reach from the starting point to reach to whatever other areas you're interested in. So these are 3 different ways of investigating these network properties. So when we applied these techniques to the diffusion spectrum imaging database from Lausanne, we found that there was no particular difference in path length if you start from the left or the right auditory cortex and you look at the path length for all other cortical nodes. However, we did see a very significant difference between left and right in the communicability index and also in this spread time measure. So in both cases, you see that there's higher communicability from the right compared to the left, and you see that there is a faster spread time from the right compared to the left. So this suggests that the right auditory cortex is more interconnected with the rest of the brain, and that greater interconnection is picked up by this communicability measure as well as by the spread time. Now, when we look at the anatomical distribution using this spread time measure, it's really quite interesting. So if you look on the left side, those 2 images on the left correspond to a seed positioned in the left auditory cortex, and the darker colors indicate faster propagation. And you can see that the faster propagation is confined more or less to the left temporal lobe, as well as a little bit in left parietal, and there's a left inferior frontal, which is interesting because it might correspond to Broca's area. You see that the spread to the right hemisphere is actually quite slow. In contrast, when you place the seed region on the right auditory cortex and look to see how much time it takes to propagate to all other regions, you see a faster spreading time throughout the entire brain, not only within the right temporal lobe itself, but also to right dorsal structures, in particular parietal and also some dorsal frontal areas. And you also see in the opposite hemisphere that there is faster spreading, again, to those more dorsal regions. So this, of course, is quite interesting and I think quite relevant to some of our hypotheses. We wanted to check whether this effect was specific to the auditory system or maybe was a more general property. And so we carried out Bratislav carried out some computations using a visual seed, and we found really no significant effects. If anything, it goes slightly in the other direction for the communicability measure. So it seems to be specific to the auditory network. And it also seems to be highly reproducible. And again, this is a theme that I think is important to show that many brain imaging results are indeed very reproducible. So when the same approach was tried with the Human Connectome data, as well as the Nathan Klein Institute database, in all 3 cases, we see no effect on the path length. We see higher levels of communicability when the seed is on the right compared to the left, and we see faster spread time when the seed is on the right compared to the left. So it seems to be a pretty good reproducible effect. So just to conclude, the idea that we have is that, first of all, auditory and motor systems are somehow preferentially interconnected. There's a lot of other evidence for that from other previous studies. And in our hands, at least, we see that it's also quite asymmetric. So it is not only the case that auditory cortex activity predicts auditory motor learning, but we also see a more sort of causal evidence when we apply TMS, engaging auditory somatomotor networks. These asymmetries are shown by both greater communicability and faster spread time from the right compared to the rest of the brain. And we think that this provides some of the substrates for this auditory motor learning. So auditory motor learning implies interactions between these systems. So if the right auditory cortex is more interconnected, it will be maybe preferentially engaged for that kind of learning. And this is actually quite consistent with some very old ideas by people like Hans-Lukas Teuber or Josephine Sems, who had proposed idea of sort of focal versus diffuse organization in the left versus the right hemisphere. The implications for plasticity, I think, are that the individual differences in these structural networks might modulate the plasticity that we see and therefore affect the behavioral outcome. And so I think this is perhaps important for those of us working in plasticity, is to take into account the initial state of the nervous system when you apply some kind of training or other intervention, so that the learning outcome is not only a property of the type of intervention that you put in, but also a consequence of the initial state, which I think can be characterized using some of these methods. Thank you all very much, and just thank you to all the people who actually did the work and the people who were involved in providing the databases that Bratislav was able to work with. Thank you.
Time for a few questions. Please go to the mic if you have one. One on the left.
Hi, Robert. Can you comment on what you think might be the origins of these asymmetries? Are they genetically predisposed?
The origins, I mean, you know, most things are, so probably yes. However, there is nothing to say that if you take two adults and you measure some property and you see differences, that that's not the outcome of some prior environmental influence that we haven't measured or we haven't been able to ascertain. It seems very likely that both genetic and epigenetic processes would influence some of these features, but that then there would be subsequent interactions with environmental influence. So it's very likely to be all of the above, and depending on which feature you're looking at, it might tend to be maybe more fixed or more stable. I like to think of it as state versus trait. So we don't know right now whether it's a trait, meaning that it's really quite stable, or is it a state that when we started the learning, that particular brain was in that particular state, but at some other time it might have been in some different state. It's a very important and interesting question, I think, for future research.
Thanks. Thank you again. Anything else? So the next speaker will be Yaniv Assaf from Tel Aviv University, who'll talk about the spatial and temporal dynamics of brain plasticity. Yaniv.
Thank you, Jason. Thank you for organizing this symposium. So we'll speak about spatial and temporal dynamics of structural plasticity in the memory domain. And to start with, let's define what is the memory domain. So the memory is the brain is like a memory device. It takes everything that we see and experience every day and just have to store it somewhere. But when you go to the neuroscientists and many to the neurobiologists, when you speak about the memory, you speak about the hippocampus. So the hippocampus is like there is in neurobiology, there is an obsession in the memory domain to the hippocampus for a very good reason, because this is like the funnel of all the memories into the brain. But using electrophysiology and neuroimaging, in recent decades, we were able to define a network of regions that interact one with another in order to produce the capabilities of memories. Well, it says next, but wait. Right? Good. So aside for the hippocampal complex that includes several subregions, the dentate drivers, the CA regions, it interacts with other regions in order to produce in the medial temporal lobe and in the cortex in order to produce the main processes of the memory domain, which is encoding of the memory and eventually retrieval of them through a very complex network of connections between all the different regions. But we shouldn't forget that the main focus of memory and learning and the main molecular process happens in the synapse regions. And eventually, no matter where the synapse is located, in the hippocampus or any other regions of this complex network, it happens in that regions. And we have to, in neuroimaging, we have to bridge the gap between things that happen on the very, very small molecular level at the synapse level to things that happen on the system level between the different regions. So this is where neuroimaging comes in. And I think so many words have been said about this seminal work of McGuire about the London taxi drivers and their lifespan changes in the hippocampus. And also about Symposium moderator Jason, the work in spatial memory-related structural changes in the hippocampus of rodents through following water maze. So there are some structural changes that happen in the brain through memory that we can get from MRI through T-bone-weighted imaging, just looking at structural changes that was shown in the memory domain, but also in other domains in the brain. And the question that comes up, what is the basis? What is the mechanism of these kind of changes? Because obviously these are massive changes that happen in the brain. Increasing the volume of cortical regions is something that is unheard of in neurobiology. And as neuroimagers, we do see that, as was shown in the previous talk, and we need to come up with a mechanism for that. So what is the mechanism of these changes? So we choose to work with diffusion imaging, as Heidi showed before, just because diffusion tensor imaging is a microstructural probe of the tissue. It could give you more finer details about what's happening in the tissue itself and get you closer. It doesn't give you any information about the synapse, obviously, but can get you closer to that kind of level of details. Not in terms of image resolution, but in terms of the information you get, just because you probe the motion of water molecules in the order of the micron scale and not just getting a big voxels of millimeters and comparing those. And that can be done either on the human level or the rodent level, and there are several indices that you can extract from diffusion imaging. Specifically in this talk, I will focus on the mean diffusivity, and for few reasons. One of them is that mean diffusivity is the most robust parameter that we can extract from diffusion imaging, and I think, broadly speaking, from MRI is a very robust parameter. The acquisition of that parameter is extremely fast. It's homogeneous across the brain, which has a lot of advantages in terms of image processing and statistical analysis, etc. The acquisition, again, is fast. You can get this parameter very, very quickly, even instantly in the new scanners with the new techniques, and it's well defined in gray matter and white matter. The problem is that the biological relation of these parameters is still vague. We need to define it. Yeah. So back a few years ago, we took these parameters in a work that was done by Sagit, Zatorre, and Hofstetter, that we looked at the changes in diffusion imaging following a spatial navigation task. So the task was to take a car in the streets of Monte Carlo, not so far from here, and drive through a track which the subject didn't know, and for 2 hours. And we compared the DTI scans before and after this task. So the protocol was to put them in the scanner, measure them with diffusion imaging, give them 2 hours of playing with the car racing game, and put them back in the MRI to see what happened. So here you can see that there is a map that is formed in the brain there's a map that is formed in the brains. can use that, in the brains of the track. They didn't see the track at all, but they do know from playing it again and again and again, eventually they can draw the track from out of the hippocampus or the memory domain and know how it looks like. And to that end, the time it takes them to complete the track just reduces. So there is a significant learning episode here. And we were able to show that you do see changes in the mean diffusivity, specifically in the memory domain. So here we see in the hippocampus, specifically more in the left hippocampus than in the right, and there are also other regions, the power hippocampus, etc. So we see reduction in mean diffusivity. And obviously, this is a very nice method. It was already replicated several times by us and other groups. So it's a very nice method in order to see which are the structural changes that are related, microstructural changes that are related to plasticity that happens in any domain, in that case specifically the memory domain. But using this method, so this is a nice method. Using this method, there are more questions that need to be asked. For example, what would be the biological mechanism of this kind of changes again? What would be the network representation of these changes? Does it stay in the same regions for a long time, or does it just moves around the brain when it's consolidated? And what is the organization? Is there an organization to the memory domain eventually? So just to go briefly through these specific topics. So what would be the mechanism, the biological mechanism behind these changes? So we use this task, a water maze task, which is more shorter than the conventional one, just performing MRI, and then a task that is completed within 2 hours. So you put the rats in the water maze. There is a hidden, if you don't know what the water maze is, so there is a hidden platform that they need to learn where it is located. The rats swim and accidentally find the platform, or you put it there, and eventually they know from swim after swim after swim where the platform is located. And once that ends, you put them back in the MRI, either immediately after that or the day later, and see what happened in the brain. So this is the learning curve. Obviously, from time to time, they experience the water pool. The time it takes them to complete the track and find the platform reduces significantly. And we do see changes in the related areas. So you can see reduction in the mean diffusivity. Reduction in diffusivity in the hippocampus, and it's well localized in specific regions. Once you do it in rodents, unlike humans, you can try and find what is the biological mechanism of that. So obviously there is a relation to the functional activity. It's very difficult to do fMRI for the memory domain in rodents. So, but we could see relation to the functional, just seeing increase in the synaptic activity in that specific region following the task. But no other markers have changed dramatically in terms of the neurons. So there is no increase in the amount of neurons after a few hours of learning, no increase in the dendritic tree, no increase in the shape of the neurons, whatever, didn't change. And we had to look to other processes that may happen. And we found very nice correlation to the amount of changes that happen on the glial cell level in the astrocytes. So the astrocytes are in green. I have no idea how to operate the movies here. So there are 2 movies here. If there is some technical guy that can, yeah. All right, so what you can see here, so these are naive rats. On the right and on the left side, you see the ones with the water maze, and you can see a huge increase in the immunoreactivity of the GFAP. GFAP is the marker of the astrocyte, meaning that these cells are more active there. Yeah, this is in a zoom of this confocal imaging. Just to see if that result is true or can be explained in a way, we took 2 ways in order to control this kind of changes. So one of them is to, so if there is increase in the synaptic activity, we can block that by using NMDA antagonists. So NMDA antagonists will just move. Yeah, can you see it? Will just go to the receptor of the glutamate on the postsynaptic terminals and will just block the activity, and therefore there will be no evidences of learning. There will be no abilities to produce LTP, etc. In order to probe for the contribution to the MRI signal by the astrocyte, there is a substance called TBOA that enables us to block the glutamate transporter antagonist. It's a glutamate transporter antagonist that enables us to block the transport of glutamate into into the astrocyte. So if there is a sensor here, a receptor here that senses that there is glutamate in the synaptic region, then it activates this channel, and then glutamate can go into the astrocyte in order to be recycled. But if you block that, then no glutamate can go in, and therefore the astrocyte is not active. So we took these two pharmacological intervention. Yeah, we'll just skip that in the benefit of time. That was first shown by the group of Mariganka School from MIT. So what we did here, we did the same water mass task, MRI. Then we did the pharmacological intervention, just injecting to the ventricles either artificial CSF for control, or AP5, which is the antagonist, or TBOA, which is the glutamate receptor blocking agent in the astrocyte, in the glial cells. They performed right after that the water maze, and then placed in the MRI again just to see what happened. Behaviorally, we could see that in the control group there is so this is the mean latency time that took them to reach the platform. So you can see in the control that we get a nice learning. The latency is about 20 seconds. The mean latency is about 20 seconds. In the AP5 group, there is no learning. They don't know where the platform is located. Even if you put them there, the next time you put them in the platform, it's like a new experience. They don't remember anything just because their synapses are blocked by the NMDA antagonist. So you can see that the latency is much higher. In the TBOA, the astrocytes are blocked, but the neurons are working. So you do get an amount of learning, actually very, very rapid learning, because there are a lot of excess of glutamate going around. You do see the learning. But the question is what's happening in the MRI. So this is the statistical map of the entire groups all together. So it's a very 3x2 ANOVA, 3 groups, 2 time points, and you can see what are the sources of that. So for example, in the dentate dryers, you see that no matter what you injected to the rats, you see the same reduction in the mean diffusivity, and this is the main area of the learning. However, the other region, CA1 in the hippocampus, you see a very nice pattern where in the control group you see a reduction in the mean diffusivity, but in TBOA it is blocked, just because the astrocyte cannot be activated in that region. And also in other regions, the entorhinal region, the entorhinal cortex hosts the same processes as well as other areas. But areas that are far away from the hippocampus or far away from the ventricles that didn't experience the pharmacological interventions show other processes. Like in the cingulate cortex, you have more activity in the TBOA, as we see, more reduction in the mean diffusivity because of the TBOA than you see in the other rats. So there is a complex pattern of changes that you can extract from this kind of experiments in order to give some kind of interpretation to the diffusion signal. But it does seem, just because you see there is no changes in the hippocampus, the main regions in the CA1 and entorhinal cortex following TBO administration, that probably most of the changes that we see are related to the astrocyte activity. All right, moving on. If you want to look about the temporal dynamic, we need to scan the rats at different time points just to see what happens in the brain when after they experience the task, what happens to their brains at a different time point. So in this experiment we've done by Shir Hofstetter, we've done several experiments. So experiment one was scanning the rats, then giving them perform the water maze. Within 2 hours, 3 sessions, 4 swims in each sessions, and then putting them immediately back to the MRI on the same day. So this is immediate working memory task. Another experiment was to do the same experiment, but to scan the rats a day later, just to see what happened to the changes that were observed on day 0. And another scan was to do the full water maze, just to see what happens when you have every day the experience, and to see what happens when the task is already fully consolidated in the brain. So short-term, immediate memory, long-term memory, consolidation. So in the short-term memory, you see a lot of structural changes that happen within. The experience is very short, few minutes of swim only. You can see many changes in there, so it's very hard to point, but you see changes. I can't get it. But you see changes in the CA regions in the hippocampus and also in the striatum as well. There is not only hippocampal learning in this task, but also stimulus-response learning. So obviously you will see striatal learning as well. But a day later, the same task, a day later, so these are the regions, so we'll just click rapidly to get over them. So hippocampus, ventral pallidum, dorsal striatum, entorhinal, etc. A day later, you can see it's the same task, completely the same task. You can see that the region starts to move in the brain. So you get the hippocampus, but you start to see also the neocortex and some sensory cortex and other areas that are involved. So obviously the same task, just a day later, overnight, you see some kind of shift of movement of these MD reductions to other regions. So it seems very dynamic and active process that even if the rats doesn't do anything, just go to sleep and wake up the next day, you see shift in this mean diffusivity to other region. So you get the sensory cortex area. And 5 days, you see a major shift of the mean diffusivity changes to more cortical regions. You do see hippocampal changes, you do get them, but most of the changes are already consolidated in the motor cortex, sensory cortex, etc. So you can define, using this method, at different time points, a network of reduction, a network of processes that happens in the memory domain, and just transfer the information through the brain in order to consolidate it and use it for later needs, if the rat would need it. At that time point, we were able to see also changes in different white matter systems like the fornix, etc. So that gives us a map of the dynamics of plasticity to that region. Things that happen in the hippocampus and the striatum just connect to other regions and then just move a day later to other regions and later on to other regions. So you can make maps like that. And the last thing I wanted to speak about is about can you use this method in order to look at spatial organization. As we heard in Heidi's talk, there is organization, for example, for the fingers in the cortex, and we do know of different organizations that we have in the brain, in the visual cortex, in the auditory cortex, in the motor cortex, etc. And the question is there a system-level organization in the hippocampus? Why wouldn't the memory domain will have a system-level organization of spatial maps or spatial organizations or whatever? And we do know that there are place cells in the hippocampus and there are time cells in the hippocampus. Nobody knows about the organization of them. There seems to be like a soup of cells that just remap every minute when you experience different spatial areas or spatial information. But that is done so that kind of information is acquired on the cellular level through electrophysiology. The question is if you can get a more holistic view, a more comprehensive view of the brain through imaging and can pin to these areas, will you get a spatial map? So in an experiment that was done by Shirov, we took 4 groups of rats. For each one of the groups, the platform was located a different quadrant, and we compared the brains of each of them. So you do get a very, very massive main effect in the hippocampus because of learning, but now you can color each pixel according to the preferentiability of the changes, meaning is there a preference for a location, meaning is there a voxels that the mean diffusivity reductions was higher for a specific location of the platform. And you do find this kind of map. So now you can color each voxel in the hippocampus based off this preference for location. And you do get like a mesh of changes. When you put it in 3D, you get this kind of map, which looks like a mess. And this is expected in a way. The hippocampus is a very complex structure, but you get like a mess of changes, and you see it's not well defined like in the visual cortex or in the auditory cortex, but you do see some kind of preference maps. And in order to explore it more in depth, we try to see what are the characteristics of these kind of changes. So one of them is just to look at the preference, either if you have preference or you don't have preference in the brain. So in these maps where you have, we call them the preference map, we just color the voxels based of the degree of preferenceability, let's call it like that. So if you have high preference for a specific location in space, it gets high number. If you have a similar preference to all locations, then you get almost 0. And you can see that there are like small areas like nodes in the hippocampus where you have very, very low preference. And these areas were very highly reproducible at many cohorts that we scanned. All of these low preferenceability areas are located in the surface between the dentate gyrus and the CA regions, like hubs of preference into the space. And when you look how the preference is located around them, you see that the preference grows in a circular manner around them, and you can get maps of the space around these hubs of no preference to spatial navigation. You can plot them around the hippocampus. Obviously, this needs to be reproduced and needs to be maybe rescanned at a different protocol that just remap the environment again and again within the same environment. But you do have the means now in order to look at that process with diffusion imaging, and it might be that the hippocampus has also some kind of spatial organization within it. So to summarize, I hope I was able to show you the diffusion MRI-based characterization of structural neuroplasticity opens new opportunities to explore the memory domain specifically. The memory domain is very complex to explore with fMRI just because the hippocampus is always active. The memory domain is always upsing and active. And looking at the structural aspects might be give more comprehensive result. The mechanism of diffusion MRI seems to be strongly related to astrocyte activity, and that should not, people say, it's not neuron, it's astrocyte. Astrocytes are a very important feature of brain function, and obviously it's very nice that we are sensitive to them in that level. And structural neuroplasticity is a dynamic process that progresses in space, in time, in the brain, and you have now the means to look at that. And finally, the diffusion MRI enables the measurements of neuroplasticity on the system level and can give us a more holistic and comprehensive characterization of the processes in way how the brain organizes itself in that sense. So just to finish, I would like to thank my team and especially Shir Hofstetter and Yael Pionkevich for their work on this project, and thank you for listening.
Time for a question or two for Yaniv. Please go to a microphone. Going once, going twice. Okay, so I will give the last talk of the session today on brain plasticity, and I will switch from the focus that Yaniv just had on imaging diffusion to imaging brain structure itself. If we can have the next talk up. There we go. So you've heard a lot of these studies described by Yaniv, by Heidi, and read about them in literature, the fact that brain structure changes due to environmental stimulation, whether it's the famous Eleanor Maguire taxi driver studies, the juggling studies, learning music, exercise, motor learning, etc. And the big question we always have is, what is it? So Yaniv addressed quite a bit of it on what are the diffusion changes, especially the short-term diffusion changes due to. We wanted to ask what are the changes in volume due to by looking at mouse models. So we wanted to start by reproducing the human data in the mouse, ask how task-dependent is it? Are we really getting a type of functional imaging if we're doing these types of short-term structure changes? Find out what we can correlate with histology to get sense of what the mechanism might be. You can infer quite a bit of mechanism from timing based on what we know about the cellular levels of plasticity, and then see if we can block that mechanism at the end to see if we can convince ourselves what it is that we're getting. So first, trying to reproduce this data in the mouse. So I'm sure all of you are quite aware of Eleanor Maguire's taxi driver study, which was really one of the first in the field to show that the brain is plastic in adults at the structural level that we can see with MRI. So we wanted to create our bus driving and taxi driving mice. And Yaniv alluded to it a bit already, but the water maze is you put a mouse into a pool, and you see a platform there. Now, the platform is actually submerged, so the mouse can't see it. Mice are dumb, but not that dumb. And after a while, we either guide them to the platform or they find it by accident, and then they begin to learn it. So this now is a mouse that's learned. It goes right to the pool. And because we're cruel and heartless scientists, at the end of the trial, we remove the platform and see if the mouse searches in the right spot. At this point, the animal hates us with a passion. We then do our imaging. In the initial studies that I'll talk about, we did it with ex vivo MRIs. We opted for high-resolution studies. After 5 days of training the mice, put them out of the scanner for 10 days, sacrifice them, and look at the brain. So this time we're scanning at between 30 to 40 micron isotropic voxel resolution after adding gadolinium to the brains. And to get the kind of throughput we need, we did this multiple mice at once in an overnight scan. So we have 16 mouse brains. Each of them sit in these individual solenoid coils. The data analyses are going to be quite known to you. They're adopted from the human imaging world, where you can start off by taking an atlas and backpropagating it to each animal in order to then ask what's the volume of the hippocampus, the volume of the dentate gyrus, etc. Or we can go to a more voxel-based, deformation-based morphometry type approach, where we take all of the scans, linearly and nonlinearly deform them to a common space, invert the deformation fields, and ask about the Jacobian determinants to get an index at every voxel of is that particular part of the brain bigger or smaller. And because mouse brains are lissencephalic, this actually works much, much better than it does for human scans. We can get to quite close to perfect matching between animals, but it's somewhat analogous to either tensor-based morphometry in humans or what you would get from voxel-based morphometry. So back to the mouse studies. When we trained these mice on 5 days of the maze and different variants of the maze, we found that we could, if we gave them a non-spatial version of the task, get about a 2% change in volume in the striatum. And if we gave them a spatial variant of the task, it's the hippocampus that grew by about 3.5% in this particular case. And looking at the more local level, areas in red are increases compared to controls, areas in blue are decreases. In both the mice that had to learn a non-spatial variant of the task, our bus mice, or that had to learn a spatial variant of the task, our taxi mice, we could see increases in volume in the dentate gyrus. In the taxi mice, and also we had additional changes in the CA1 and CA2 areas of the hippocampus, whereas in the bus-driving mice, we changed the striatum as well. It's more of a stimulus response task. And if we just compare the exercise-matched versions of the task, the non-spatial mice had this fairly focal CA1 area increase in the hippocampus, whereas there was an increase in the lateralized right striatum in the non-spatial variant of the task. Now, quite striking that it's so lateralized in the striatum. Don't know why. It'll come up again later, so it's somewhat reproducible, but as an unknown as to why there would be any kind of laterality in the striatum. So then, are these types of changes task-dependent? We started first by trying to just reproduce the water maze task, but using a radial arm maze. So similar idea, mouse has to use spatial cues in order to identify where the target is. But in this case, it starts in the center of the platform. There's 8 arms radiating outwards, and 4 of them are baited, so it's a food reward. And the mouse eventually learns which of the 4 arms are baited as it does the same task over 5 days. If we then look at it, we find the exact same changes in the CA1 that we saw before on the water maze. Interestingly, also these rightward decreases in the striatum if they learned the spatial variant of this 4 and 8 radial arm maze task. If instead we put the mice on a rotor rod, so this is really a skilled motor task, the mice are placed on this rod, it's a little bit above the ground, so they want to stay on it, and then the rod begins to spin faster and faster, and we time how long the mouse can keep on it. They do quite poorly in the first day, and then they learn to do better and better at this motor adaptation. In that particular case, we also see changes in the hippocampus, more in CA3 as well as dentate gyrus in this case, as well as some increases in volume in the motor and sensory motor cortex, and some decreases in cerebellar volume. Lastly, the sort of omnibus task, we put mice in an enriched environment, which you see here. So you have these mice having wheels in their cage, they have mazes, they love wheels. Mice will run and run and run if you give them a chance, even more than one at once. And ask them what does these significant changes in activity and what they can do in terms of their home environment, how does that change the brain? In that particular case, we again see strong dentate gyrus and CA3 changes in the hippocampus, as well as some visual cortex, retrosplenial cortex, and a little bit of sensory motor cortex as well. So in short summary, so far from the task dependence, we do see that there are some task dependence in these volume changes, and these were all tasks that were done over a period of about 2 weeks or so of training and or living in an enriched environment. The hippocampus almost always alters. So you give a mouse any kind of task, whether it's particularly designed to be spatial, whether it's more of a motor task, we see volume changes in the hippocampus. It's the extrahippocampal variants of it, so the striatum and the cortex, that differs depending on what the type of task is. We then went back to the initial study of the mice on the water maze and correlated what we found with histology. And very similar to what Yaniv showed before, there were no changes in terms of neurons, no changes in astrocyte volume in this particular case, no changes in neurogenesis. But we did find a somewhat noisy but still significant correlation with GAP43, which is an axonal growth cone marker. So effectively, there's something changing at this neuropil level in terms of the number of synapses and dendritic branching, axonal branching, that correlates with the volume changes that we see on MRI. There's a lot of information, if we believe the story, that we're starting to see these changes at the neuropil level with changes at the dendritic spine axonal bouton that we can get from just looking at the timing of these types of events. So very rough overview of, very superficial overview of brain plasticity. You start by having neurons firing, which happens within seconds and milliseconds of starting a particular task. And then over a period of minutes lasting into hours, this initial firing potentiates, I get long-term potentiation, long-term depression, in order to keep the learning going. Long-term potentiation depression can last for up to 3 or 4 hours without any other changes. And at the same time as the cells are potentiated, you also have this astrocytic swelling that Yaniv showed so elegantly and seems to really be a source of the diffusion signal that we're seeing. To extend plasticity beyond the first minutes or the first few hours, you start to need protein synthesis. So effectively, you've gone as far as you can by simply changing synaptic strength. Now you have to start forming new synapses, generate new growth cones potentially, new dendrites, etc., all pretty well established in the molecular plasticity field. And this takes it from minutes into hours and hours into days, and eventually results in changes at the number of dendritic spines that you have, number of axonal boutons that you have, potentially even larger scale reorganization at the dendritic tree level. So if we think that the changes we're seeing are at the protein synthesis dependent hours to days level, then we should see changes in the brain that really occur after a day or two and grow a bit bigger as time goes on. So we started at this point moving on to looking at in vivo MRI. So we did various time courses, and we did manganese enhanced magnetic resonance imaging, because we always do it more than once. We do 7 mice at once in this case, and we're scanning at 90 micron resolution. If you look at just the enriched environment showed before, with mice in it for 24 hours, we see these volume increases in the striatum, volume increases in the dentate gyrus, as well as some decreases in volume in the cerebellum. So in some ways, it looks quite similar to the very short-term changes in diffusion that Yaniv showed in the talk just before me. If we look at just the volume of the hippocampus over time, so there are 2 scans done before the training started, or before we put them into the enriched environment. We averaged them, took them as the baseline signal, and then looked at growth over the next while. You can see that there's an initial hint of growth in the first 24 to 48 hours, and then gets to be significantly different in volume over 1 to 2 weeks. And if I just zoom a bit in on this early time point, already trends are apparent at 24 hours, but not significant. And then they reach very borderline trend significant levels at 48 hours before accelerating as time goes on. The synaptic level also can take us away a bit from the pure task-based type of designs and looking at a more naturalistic plasticity level, where if you look at the rat estrus cycle, it's been quite well established that the number of dendritic spines vary across the cycle itself. And that's been shown a bit less reproducibly, but also in the mouse. So effectively, the rat and mouse estrus cycle lasts 4 to 6 days. And during that period, you have these fairly wild fluctuations in terms of estradiol and progesterone. And there's many, many receptors, estradiol receptors in the hippocampus. So you have high estradiol during proestrus, it drops during estrus, and then towards diestrus, progesterone rises again. And you have multiple synaptic markers that are shown in those bar graphs up top from the literature that change at the same time. If we look at hippocampal volume in this first study ex vivo across mice across the estrus cycle, we can see that day by day you have changes of about 1 to 2% in hippocampal volume, with the largest volume being during proestrus. And then as estradiol and progesterone are low, there's a drop in volume, then increases again towards diestrus. And if we follow the same mice and scan them one day after the other, you can see these changes as well, now expressed as percent change, with a drop in volume of the hippocampus from one day to the next as you go from proestrus to diestrus. Then it slow increases again over the next 2 or 3 days as you go back towards diestrus and proestrus. So hints that the plasticity mechanisms we're seeing are indeed noticeable at the synaptic level, even in this naturalistic plasticity study where we know that synapses are changing due to fluctuating hormones. So then we wanted to block this particular mechanism, going back to the task-based types of design. And from the literature, we thought that one of the key mechanisms might be the CREB transcription factor. So CREB is particularly important in turning long-term potentiation into gene expression, so protein synthesis, et cetera, and helps to stabilize memory. So if you have CREB mutant mice, then you do not get very good hippocampal and amygdala-dependent learning. So we went to mice which had 2 of these particular isoforms of CREB knocked out, so they have very little functional CREB, and we used both homozygous and heterozygous mutants to study the effects on brain plasticity. So this is just the learning curve on the water maze, where in the blue and the green line you have in the red line you have the wild types, in the blue you have the heterozygous knockouts. You can see that they learn quite efficiently over the 6 days, 5 trials per day of the water maze. Whereas in green, you have the homozygous knockouts. They do learn. They get to close to the same levels by day 6, but they clearly learn much slower. If we then look at the hippocampal volume after this particular task, in red, you can see the CREB mice in the control condition or in the spatial condition. There's a volume increase of about 2% or so, significantly different. In blue, you have the heterozygotes, which have no volume change whatsoever, depending on whether they were in the water maze training condition or not. And interestingly, and somewhat confusingly, in the homozygous knockouts, you have some trends towards a change in hippocampal volume as well, though this isn't significant. So we're a bit unsure what to do with this. We're currently trying to replicate the study in a radial arm maze instead to see if we can indeed see this difference, where it appears to us that learning is both hindered by the knockout of loss of CREB, both heterozygous and homozygous, and it seems to inhibit volume change, but we want to validate that. But if we put these mice into the enriched environment, there's a very clear and strong trend that by the end of it, by 2 weeks, volume of the hippocampus grows with enrichment completely independently of the particular Krebs isoform that the mice have. So wild types, heterozygous, and homozygous have the same amount of volume change. There's some trend towards the very early changes, 24, 48 hours, being significantly different in the wild type versus the two mutants, but again, only borderline significance level. So what we think is happening is that there's really different mechanisms in terms of plasticity, where exercise changes the brain, changes the hippocampus, almost independently of these kind of mechanisms that we're talking about learning, whereas in learning, you might be able to inhibit it by going at the long-term potentiation type effects, in this case with Krebs mutants. Again, somewhat similar to the designs that Yaniv was showing you early on, where no matter what you did, the dentate gyres grew in volume, where some of the more lateral CA areas were more learning dependent, which seems to be what we're seeing as well, but again, studies still ongoing. So I'm going to finish up this particular talk. We've shown you that you can indeed reproduce these kind of changes in the mouse and in the rat. And of course, by doing so, you can get access to the different mechanistic studies that are very, very hard to do in human subjects. It seems to be task dependent, as long as you're looking outside of the hippocampus, but the hippocampus itself changes almost no matter what kind of task you put the mouse into, especially if there's some kind of exercise component to it. It correlated with GAP43, which is an axonal growth cone marker, so it makes us think that the changes are really happening at the synaptic, dendritic, and axonal level. Changes occur over a day and get bigger as time goes on. Uncertain, based on the knockout studies, whether we really have different mechanisms that are happening at the same time, where the early changes are quite learning dependent, potentially dependent on the Krebs transcription factor, later changes being more dependent on exercise, independent of Krebs itself. So with that, thank you for your attention, and thank you especially to Dulcie Vouston, who also has a poster here, who's done most of the work, along with Jan Scholzen, Rylan Allman Grant. We have time for a question or two.
Very nice talk. I just bring a question into attention. Since we know the functional training and learning bring up the blood flow. So blood flow has slightly different T1 than tissue. So when the tissue is get more perfused, the tissue value will change. That's the first point. The second point is when the tissue is more perfused, that will automatically bring up the tissue volume. So in this year, we have abstract 1916 showing that short-term blood flow can actually introduce artificial volume change. So I'm wondering whether this apply to this to your experiment or not, or did you measure the blood flow at the same time?
So I showed you both ex vivo and in vivo data, and I can assure you there wasn't much blood flow in the ex vivo mice. So I definitely think it's a valuable confound or artifact to look for, especially in some of the in vivo data that we have. But at least in our case, given that we've shown very much the same finding using two different sequences, ex vivo and in vivo, I'm pretty sure that at least a component of it is exercise independent in this case. But definitely thank you for bringing that up. There's many confounds here we all have to deal with, try to figure out what these changes are due to.
Yes, actually, I'm not saying all of the effects are contributed by blood flow, but there may be, some part of it.
Right.
Thank you.
There seem to be no further questions, so thank you again to Robert, Heidi, and Yaniv, and thank you everybody for staying for the session.